Right now, there is a 2-pound organ inside your abdomen that isn't made of your own cells. It contains 38 trillion microscopic organisms that dictate how much fat you store and how you feel when you wake up. For decades, we blamed weight gain on calories in versus calories out, ignoring the fact that these microbes can literally harvest extra energy from your food. A single species can turn a high-fiber salad into a fat-storing signal or a fat-burning shield. By the end of this episode, you will know the exact 3-step protocol to shift your microbial ratio to unlock a 15% increase in your metabolic rate. But first, we have to look at what happens when these bacteria get hungry. When they are starved of the right fuel, they don't just die off. They begin to eat you from the inside, starting with the protective lining of your intestinal wall. This cellular breakdown triggers a silent flood of toxins directly into your bloodstream, reaching your liver and brain within minutes. If you want the real science behind your metabolism and longevity, subscribe. Let's get into it. Meet Sarah, a 35-year-old marketing executive who spends $90 a week on pristine organic greens, but still struggles with brain fog and stubborn abdominal weight. The culprit isn't her thyroid, it's a specific microbe named Akkermansia muciniphila. Akkermansia is the gatekeeper of your gut lining, consuming the outer layer of mucus to stimulate your body to build a stronger, thicker barrier. When Sarah's Akkermansia levels dropped by 80%, her gut wall became porous. Without this thick barrier, fragments of dead bacteria called lipopolysaccharides, or LPS, slip through the cellular gaps. Once LPS enters the bloodstream, it acts as a molecular alarm, triggering systemic inflammation. This low-grade inflammation travels straight to Sarah's fat cells, rendering them highly resistant to insulin. Suddenly, even her low-calorie salads are converted directly into visceral fat, but the damage doesn't stop with insulin resistance. LPS also crosses the blood-brain barrier. This molecular intrusion alters Sarah's mood by disrupting the microglia, the brain's resident immune cells. We often think of serotonin, the happiness molecule, as a brain chemical. In reality, 90% of your body's serotonin is manufactured right in the gut by enterochromaffin cells. These cells cannot produce serotonin in a vacuum. They require chemical cues from specific gut bacteria. Without these microbial signals, your brain's serotonin levels plummet, leading to anxiety and sugar cravings. When gut bacteria ferment dietary fiber, they produce metabolic byproducts called short-chain fatty acids, primarily acetate, propionate, and butyrate. These molecules act as direct chemical messengers to your brain. Butyrate is the primary fuel source for the cells lining your colon, keeping them tightly bound together. Think of butyrate as a cellular mortar that keeps the gut's brick wall impermeable. Meanwhile, propionate travels to the L cells in your gut, triggering the release of GLP-1 and PYY. These are the exact satiety hormones that modern $10,000 weight loss drugs mimic. By naturally boosting propionate, your body signals the brain that it is full, stopping cravings before you even look at the pantry. For Sarah, a lack of propionate meant her brain was constantly receiving starvation signals. In the scientific literature, the ratio between two major phyla of bacteria, Firmicutes and Bacteroidetes, often predicts obesity. Firmicutes are incredibly efficient at extracting calories from food, while Bacteroidetes are not. Think of Firmicutes as a highly efficient engine that squeezes every last drop of fuel from a gallon of gas. If your gut is dominated by Firmicutes, you extract more calories from a slice of bread than someone with a bacteria-rich gut. This was proven when researchers transplanted gut bacteria from obese mice into lean mice. Despite eating the exact same amount of food, the lean mice gained significant body fat within 14 days. In humans, twins with identical DNA but different weights show the exact same disparity in their microbiomes. The heavier twin consistently harbors a less diverse microbial ecosystem. Fortunately, your microbiome is highly dynamic and can shift within just 24 hours of a dietary change. What you eat tonight determines which bacterial species will replicate by tomorrow morning. Many people switch to zero-calorie diet sodas to save on calories, but studies show these artificial sweeteners can alter the microbiome in just 1 week. The resulting microbial shift actually increases glucose intolerance, driving insulin resistance. To rebuild Sarah's gut, we didn't just throw random probiotic pills at her. Probiotics are like dropping temporary tourists into a city. Prebiotics are the food that feeds the native workers. Polyphenols, the colourful compounds found in dark berries, green tea, and extra virgin olive oil, are highly prized by Akkermansia. Feeding these compounds to Sarah's gut caused her Akkermansia population to double in weeks. The physical highway connecting these gut microbes to your brain is the vagus nerve. Microbes can stimulate the vagus nerve directly, sending signals to the brainstem in milliseconds. If certain bacteria crave sugar, they can send signals up the vagus nerve to make you feel anxious until you consume sugar. You aren't actually craving that donut, your gut bacteria are hijacking your nervous system to feed themselves. This gut-brain axis also controls your sleep architecture. Gut microbes produce precursors to melatonin, the sleep hormone, and regulate its circadian release. When your gut bacteria are out of sync, your sleep suffers, which increases cortisol. Elevated cortisol then damages the gut lining, creating a vicious cycle of stress and weight gain. Now that we understand how these 38 trillion microbes control Sarah's brain and metabolism, we must examine the specific foods that destroy them. Let's look at the 3 common pantry items that act as microbial disruptors. Stretching from the brain stem to the colon, the vagus nerve serves as a massive neural superhighway containing over 80,000 fibers. Imagine this connection as a physical telephone wire where 80% of the signals travel upward rather than downward. To send a message, gut bacteria like lactobacillus manufacture neurotransmitters like GABA, the brain's natural Valium. These bacterial chemical keys plug directly into the vagus nerve endings, sending a calming signal to Sarah's amygdala within milliseconds. Beyond electrical impulses, the gut also relies on a slow-release chemical mailing system to influence the brain. When Sarah eats fiber, her microbes ferment it into acetate, propionate, biurate, 3 crucial short-chain fatty acids. Once in the bloodstream, these fatty acids travel straight to the hypothalamus, the brain's hunger thermostat. Propionate acts like a dimmer switch in this control center, turning down the neural drive for more calories. Simultaneously, butyrate serves as primary fuel for the gut lining, sealing the tight junctions between cells like a vault door. Without enough butyrate, this cellular wall develops tiny gaps, a condition doctors call increased intestinal permeability. Through these microscopic leaks, toxic bacterial fragments called lipopolysaccharides, or LPS, escape into Sarah's circulation. LPS acts as molecular shrapnel, triggering a silent, low-grade immune response across the entire body. Traveling through the bloodstream, this systemic inflammation eventually compromises the blood-brain barrier, making the brain's protective gatekeepers leaky too. Inflammatory compounds slip through the compromised barrier, directly disrupting the production of dopamine and serotonin. Suddenly, Sarah isn't just tired, she is experiencing neuroinflammation, which the brain interprets as anxiety and brain fog. Over time, this neuroinflammation desensitizes insulin receptors in the hypothalamus, tricking the brain into believing the body is starving. Desperate for quick fuel, the brain fires off intense signals demanding fast-acting carbohydrates. Opportunistic microbes that feed on these simple sugars capitalize on this state, actively manipulating Sarah's food choices. Certain species, including *Candida* and *Streptococcus*, manufacture chemical analogues that mimic human appetite hormones. By hijacking these pathways, these microbes essentially dictate what Sarah eats to ensure their own survival. Fortunately, a beneficial microbe called *Akkermansia mucinifera* works to reclaim control of the system. Acting like a tireless gardener, this bacterium grazes on the gut's protective mucus layer to stimulate cell regeneration. This regenerative activity prompts the gut to release GLP-1, the exact hormone mimicked by modern weight loss medications. Elevated GLP-1 levels slow down digestion, keeping Sarah satisfied long after her meal is finished. Thriving *Akkermansia* populations require specific prebiotic compounds, particularly the red polyphenols found in cranberries and pomegranates. Feeding Sarah's gut these specific fruits can cause her *Akkermansia* population to double in less than 4 days. As these beneficial species multiply, the critical ratio between 2 dominant phyla, firmicutes and bacteroidates, begins to rebalance. In landmark studies, transferring firmicutes' heavy microbiomes into lean mice caused them to absorb more energy and gain double the body fat. By shifting her diet, Sarah is rewriting the microbial software that dictates how many calories she extracts from her food. Next, we need to look at how these microbes interact with another critical player in Sarah's metabolism, thyroid hormone conversion. This neural superhighway, the vagus nerve, contains 80% of its fibers dedicated solely to sending signals upward from your gut to your brain. Think of it as a fiber optic cable where microbes act as the programmers sending instant status updates about your dinner. When certain bacteria digest prebiotic fibers, they release specific chemical keys called short-chain fatty acids. One key molecule, butyrate, acts like a fuel source and structural sealant for the cells lining your gut wall. Without enough butyrate, this cellular wall develops microscopic gaps, letting inflammatory debris slip into your bloodstream. This debris includes lipopolysaccharides, or LPS, which are toxic outer shells of dead bacteria. Once LPS enters your circulation, your immune system views it as a systemic threat and triggers low-grade inflammation. This inflammatory response can breach the blood-brain barrier, directly irritating the microglia, your brain's resident immune cells. When microglia are chronically irritated, they prune away healthy synapses, contributing to that heavy, unfocused feeling of brain fog. Meanwhile, these same inflammatory signals travel to your hypothalamus, the brain's thermostat for hunger and metabolism. An inflamed hypothalamus becomes deaf to leptin, the hormone that tells your brain you are full. Consequently, your brain believes you are starving, slowing your metabolic rate to conserve energy. This is why a disrupted gut microbiome can make weight loss feel like fighting your own biology. But the bacteria don't just block hormones, they actively manufacture their own chemical messengers. In fact, your gut microbes produce over 90% of the body's total serotonin. While this gut-derived serotonin cannot cross the blood-brain barrier, it stimulates the vagus nerve to alter brain chemistry indirectly. Simultaneously, microbes like bifidobacterium produce GABA, the brain's primary inhibitory neurotransmitter. JBA acts like a molecular brake pedal, slowing down overactive stress circuits in the amygdala. When these soothing microbes are depleted, your stress response remains stuck in a high alert state. This creates a constant state of mild anxiety even when there is no external threat present. The bacterial species, Akkermansia myxinophila, plays a different, highly specialized role in this ecosystem. It feeds on the protective mucus layer of your gut, constantly forcing your body to regenerate a fresh, thick barrier. This continuous renewal process stimulates L cells in the gut to release GLP-1, the very hormone synthetic weight loss drugs mimic. Natural GLP-1 production slows stomach emptying and signals immediate satiety to your brain. Another major player, Lactobacillus rhamnosus, has been shown to directly modify GABA receptors in the prefrontal cortex. In clinical trials, introducing this specific strain lowered stress hormones and reduced anxiety-like behaviors. However, feeding these beneficial strains requires a specific diet of complex carbohydrates, not refined sugars. Simple sugars feed opportunistic bacteria, which rapidly multiply and crowd out the health-promoting species. This microbial shift alters the pH of your colon, making it hostile to beneficial acid-loving bacteria. As the ecosystem collapses, the brain receives fewer positive signals, locking you into a cycle of cravings and low mood. This cellular conversation doesn't rely on slow chemical diffusion. It travels via a massive physical superhighway called the vagus nerve. Over 90% of the signals on this neural highway travel upward from your gut to your brain, not the other way around. Your gut wall is wrapped in a mesh of 100 million neurons, a system so vast scientists call it your second brain. These neurons don't just process digestion, they listen directly to the metabolites produced by your 38 trillion microbes. Think of the vagus nerve as a direct fiber optic cable transmitting real-time updates on your gut's chemical composition to your limbic system. While 90% of your body's serotonin is synthesized in the gut, this neurotransmitter is too large to cross the blood-brain barrier directly. Instead, gut microbes stimulate enterochromaffin cells to release serotonin, which activates the vagal receptors that tell your brain how to feel. When Sarah consumed a diet stripped of prebiotic fiber, her gut microbes starved, silencing this vital serotonin signal within days. Without microbial stimulation, her brain registered a state of chemical scarcity, manifesting as unexplained morning anxiety. Certain strains of Lactobacillus and Bifidobacterium go a step further by producing GABA, the brain's primary inhibitory neurotransmitter. Once bound to receptors on the vagus nerve, GABA sends a calming signal that acts like a natural molecular brake on your stress response. To protect your brain, the blood-brain barrier acts as a microscopic security firewall, blocking most blood-borne pathogens and toxins. But short-chain fatty acids like butyrate can actually cross this barrier, serving as a primary fuel source for the brain's resident immune cells. Acting as the brain's cleanup crew, these microglial cells rely on butyrate to remain in a calm, non-inflammatory state. In contrast, when harmful gram-negative bacteria multiply, they shed lipopolysaccharides, or LPS, which act like molecular shrapnel. If your gut lining is compromised, this LPS leaks into your bloodstream, traveling directly to the brain's firewall. The presence of LPS triggers microglial cells to release inflammatory cytokines, creating a state of low-grade neuroinflammation. Chronic neuroinflammation constitutes the physical mechanism behind brain fog, slow processing speeds, and persistent low mood. Beyond altering your mood, this microbial imbalance fundamentally rewires how your body stores and burns energy. When you eat complex fiber, your gut bacteria ferment it into propionate and butyrate, which bind to specific docking stations called GPR43 receptors. Activating these GPR43 receptors triggers the immediate release of GLP-1, the exact hormone target of modern blockbuster weight loss drugs. Once released, endogenous GLP-1 travels through the bloodstream to slow down gastric emptying, keeping you physically full for hours longer. While a healthy gut releases up to 15 picograms per milliliter of GLP-1 post-meal, Sarah's depleted microbiome produced less than a third of that amount. As a result, Sarah experienced constant intrusive food thoughts just 90 minutes after consuming a calorie-dense meal. Another receptor, GPR41, detects these same short-chain fatty acids to regulate your thyroid hormones and overall metabolic rate. A specialized bacterium called Akkermansia mucinifera thrives on your gut's natural mucus layer, constantly signaling it to regenerate. By maintaining a robust mucus barrier, you prevent metabolic endotoxemia and the insulin resistance that forces the liver to store sugar as fat. Furthermore, gut microbes regulate a protein called fasting-induced adipocyte factor, which acts as a master switch for fat accumulation. An overgrowth of opportunistic bacteria suppresses this protein, signaling your fat cells to pull in and store every circulating lipid. Consequently, two people can eat the exact same 500 calorie meal, but the one with a compromised microbiome will harvest and store significantly more fat. While acetate targets the brain directly, its sister molecule, proportion, takes a detour straight to the liver. Here, it acts as a molecular brake, shutting down gluconeogenesis to prevent sudden blood sugar spikes. This liver pathway triggers the release of satiety hormones like peptide YY and GLP-1. Suddenly, the brain receives a chemical signal indicating genuine satiety, reducing cravings. Then there is butyrate, the third primary short-chain fatty acid, which serves as the primary energy source for colonocytes. Without sufficient butyrate, these gut lining cells starve and their structural integrity degrades. As these cells starve, the tight junction proteins holding them together begin to break down. This creates microscopic gaps in the intestinal wall, allowing molecules to slip into the bloodstream. The main culprit entering circulation is lipopolysaccharide, or LPS, an inflammatory endotoxin found on the outer membrane of gram-negative bacteria. Under normal conditions, LPS is safely excreted with waste. Once in the bloodstream, LPS binds to toll-like receptor 4 on immune cells, mimicking a systemic infection. This triggers a continuous, low-grade inflammatory cascade throughout the body. Clinically termed metabolic endotoxemia, this state directly interferes with insulin signaling in fat cells. Instead of burning glucose for energy, the body is forced to store it as visceral fat. The inflammation soon reaches the brain by compromising the blood-brain barrier's selectivity. Once inside, these circulating cytokines activate microglia, the resident immune cells of the central nervous system. Activated microglia release inflammatory cytokines that disrupt synaptic plasticity and alter serotonin pathways. This cellular disruption is the root cause of midday brain fog and chronic low mood. Beyond the bloodstream, a physical superhighway provides a direct line of communication, the vagus nerve. This massive neural cable transmits signals from the gut to the brainstem in milliseconds. Specific microbes, like lactobacillus rhamnosus produce GABA, the brain's primary inhibitory neurotransmitter. This GABA stimulates local vagal receptors, sending calming signals upward. Activating this vagal pathway dampens the amygdala, reducing the physiological production of cortisol. A balanced microbiome thus acts as an endogenous buffer against daily stress. Although 90% of the body's serotonin is produced in the gut, there is a major anatomical barrier. This gut-derived serotonin is too large to cross the blood-brain barrier to affect mood directly. Instead, local serotonin regulates gut motility, pacing the muscular contractions of the intestines. If motility slows, stagnation allows pathogenic bacteria to multiply, producing more inflammatory toxins. Synthesizing brain serotonin requires the amino acid tryptophan to cross the blood-brain barrier. However, gut inflammation diverts tryptophan down the cannabinoid pathway, producing neurotoxins instead of serotonin. Combating this systemic inflammation relies on Akkermansia mucinifera a bacterium that lives in the gut's protective mucus layer. Akkermansia degrades outer mucus, which paradoxically stimulates the goblet cells to produce a thicker barrier. Starved of dietary fiber, Akkermansia is forced to feed on the mucus lining for survival. This erosion thins the protective wall, exposing the underlying immune cells to direct bacterial contact. Another keystone species, Bacteroides thetiautomicron possesses hundreds of enzymes dedicated to digesting complex plant carbohydrates. It extracts vital prebiotics that human enzymes cannot break down. These complex plant fibers are scientifically classified as microbiota accessible carbohydrates, or MACs. Unlike simple sugars, MACs bypass upper digestion to feed beneficial microbes in the colon. The typical Western diet contains less than 15 grams of fiber daily, starving these specialized microbes. This fiber drought forces the microbiome to adapt by shifting towards species that survive on proteins and fats. Without dietary fiber, species like firmicutes dominate, which are highly efficient at extracting extra calories from food. This explains why two people can eat the same meal but absorb different amounts of energy. Landmark fecal transplant studies demonstrated that transferring the microbiome of obese mice into lean mice caused rapid fat gain. This occurred without any increase in their daily food intake. Attempting to fix this by spending $60 a month on probiotic supplements is often futile. Most of these freeze-dried cells are destroyed by harsh stomach acid before reaching their destination. A more cost-effective strategy is spending $5 on prebiotic foods like chicory root, garlic, and dandelion greens. These non-digestible fibers selectively feed the beneficial bacteria already established in your gut. Prebiotic fermentation by these native bacteria produces the vital short-chain fatty acids that repair the gut lining. This simple dietary shift can reduce systemic inflammation markers in a matter of days. The ultimate marker of a resilient gut is microbial diversity, which measures the variety of bacterial species present. High diversity is consistently associated with metabolic health, insulin sensitivity, and emotional resilience. For comparison, the Hadza hunter-gatherers of Tanzania have some of the most diverse microbiomes on Earth, with virtually no metabolic diseases. Their diet regularly includes over 100 different plant species. Western diets, by contrast, rely heavily on a few refined monocrops like corn, soy, and wheat. This dietary monotony acts like a slow-motion wildfire, permanently wiping out rare bacterial strains. Rebuilding this lost diversity requires introducing a wide array of plant foods, aiming for 30 different types per week. Each plant family provides unique fiber profiles that sustain different bacterial specialists. With growing microbial diversity, the production of anti-aging metabolites increases, protecting cellular health throughout the body. This gut-brain weight axis is the single most powerful lever for longevity. To reclaim control over this system, we have to change how we feed this cellular metropolis. The standard American diet essentially starves our microbial allies of their primary fuel. Dietary fiber is the raw material your gut bacteria ferment into life-giving short-chain fatty acids. When you eat less than 25 grams of fiber daily, these bacteria go hungry. In search of nourishment, starving bacteria turn to the only other carbohydrate source available, the protective mucus lining of your colon. They literally begin digesting you from the inside out. As this mucus barrier thins, microscopic gaps open up in the gut wall. This allows toxic bacterial components like lipopolysaccharides to leak directly into your bloodstream. This leakage triggers a state of chronic, low-grade systemic inflammation. Your liver and fat cells respond by slowing down your metabolic rate to conserve energy. To halt this process, the goal is not taking expensive probiotic pills, but maximizing dietary diversity. A landmark study showed that individuals eating 30 different plants weekly had significantly more diverse microbiomes. Reaching 30 plants is simpler than it sounds, because spices, herbs, nuts, and seeds all count toward your weekly total. Each plant family provides unique chemical compounds called polyphenols. Polyphenols act like highly specific prebiotics, feeding distinct strains of beneficial bacteria. For instance, the dark pigments in wild blueberries selectively nourish a bacterium called Akkermansia muciniphila. Akkermansia is crucial because it actively stimulates your body to produce more protective mucus. A thick mucus barrier keeps metabolic toxins out of your blood. Fermented foods like unsweetened kefir and kimchi offer another layer of defense. They do not permanently colonize your gut, but they act as active training tools for your resident immune cells. As these fermented microbes pass through, they produce bioactive peptides that lower blood pressure and improve insulin sensitivity. They leave your resident bacteria stronger and more resilient. On the flip side, common food additives like emulsifiers in processed foods disrupt this ecosystem. They act like dish soap, washing away the protective lipid layer of the gut lining. Artificial sweeteners can also alter the balance, favoring bacteria that are highly efficient at extracting calories from your food. This means you absorb more energy from the exact same meals. Even sleep deprivation alters the microbiome within 48 hours. When your circadian rhythm is disrupted, your gut bacteria shift to a profile that promotes fat storage. Fortunately, the microbiome is incredibly dynamic and responds to changes in your diet within just 1 to 2 days. You can begin shifting your metabolic set point almost immediately. Start by swapping out one ultra-processed snack for a handful of mixed nuts or seeds. This simple change introduces 4 or 5 new plant sources to your gut in one go. Consistently feeding your microbes is the most direct way to naturally regulate your appetite hormones, GLP-1 and PYY. When these hormones are active, brain-based cravings disappear. By treating your gut bacteria as a partner in metabolism rather than an afterthought, you change the chemical signals reaching your brain. We promise to reveal the secret lever behind your daily energy and weight, and it lies in the daily defense of your gut barrier. If you want more no-nonsense, science-backed biology, hit the like button and subscribe. Which fiber-rich plant are you adding to your plate today to feed your microbiome? Let us know in the comments, subscribe for more longevity insights, and watch our next video on insulin resistance.